Hello everyone and welcome back to my YouTube channel. Today I will be teaching the chapter Photosynthesis in Higher Plants from class 11 Botany, which many students find hard to understand. So let's get into it. Photosynthesis is the most important process on Earth. Photosynthesis is the process by which green plants use light energy to synthesize food in form of glucose while releasing oxygen in the atmosphere. It is the foundation of life on Earth as it provides energy and oxygen for all living organisms. Green plants capture solar energy and store it as chemical energy in carbohydrates. This energy is then made available to all other organisms through the food chain. Our understanding of photosynthesis has developed over centuries. Early experiments by Ingen, Hirsch, and Sachs revealed that sunlight, chlorophyll, and carbon dioxide are essential in the process. In higher plants, which we will be dealing with today, photosynthesis takes place inside the chloroplast. The chloroplast contains pigments like chlorophyll, which absorbs light energy and is a site for both the light reaction and the Calvin cycle, which you will learn soon. From these studies, we know that photosynthesis is a physicochemical process where light energy is converted into chemical energy. Carbon dioxide and water are used to synthesize carbohydrates, and oxygen is released as a byproduct. Our understanding of photosynthesis has been built over centuries through the work of several scientists. Joseph Priestley discovered that plants release a substance that supports combustion, which we now know as oxygen. He showed us that plants restore air that has been injured by burning candles or by animals. Jan Ingenhusch demonstrated that sunlight is essential for oxygen production and that it occurs only in green plant paths. Julius von Sachs showed that green plants produce glucose during photosynthesis, which is stored as starch. Theodor Wilhelm Engelmann used a prism to split light into different wavelengths and found that oxygen is released most in the blue and red regions of the spectrum, providing that these wavelengths are most effective for photosynthesis. By the mid-19th century, scientists knew that plants used light to make carbohydrates from carbon dioxide from water. An early empirical representation of the process was written as CO2 plus H2O gives CH2O plus O2. Here, CH2O represents a simple carbohydrate unit. This equation summarizes the net transformation but does not show the many steps involved. Cornelius van Niel proposed generalized equation from studies on bacteria. 2H2A plus CO2 gives CH2O plus H2O plus 2A. In this scheme, H2A is a hydrogen donor. For many bacteria, the donor is hydrogen sulfide and the oxidized product is sulfur. For green plants, the hydrogen donor is water. Van Niel's insight implied that oxygen evolved by green plants comes from water, not carbon dioxide. The common use balanced equation combining the multi-step process is 6CO2 plus 6H2O in presence of light gives C6H12O6 plus 6O2. This is a summary of many reactions, the light-driven steps that produce ATP and NADPH and the biosynthetic steps that fix CO2 to form sugars. The idea that oxygen comes from water was later confirmed using isotopic experiments. Isotopic experiments referring to radioactive or heavy oxygen isotopes. When water labeled with an oxygen isotope was supplied, the evolved oxygen contained that label, proving water is the source of the oxygen released. Most of the action takes place in the green parts of the plant. Inside each leaf there are mesophyll cells. Inside these cells are thousands of tiny green factories called chloroplasts. Each chloroplast has two main working zones. The membrane system, that is stacks called grana, connected by stroma lamellae. The stroma, which is a fluid-filled space where sugar is made. In the grana, light energy from the sun is captured and used to make energy-rich molecules that are ATP and NADPH. These are called light reactions because they run directly on sunlight. Next, in the stroma, these molecules power a set of enzyme reactions that build sugars from carbon dioxide. This is the Calvin cycle, also called the dark reactions, not because they happen in the night, but because they don't directly need sunlight. So photosynthesis takes place inside chloroplasts in two stages. First is light reactions in the grana, and the second is the sugar-making reactions inside the stroma. When you look at a plant, you see green, but did you know that green isn't the only color hiding inside the leaf? As we learn that with each leaf there are tiny structures called chloroplast. Within them live special molecules called pigments. These pigments absorb sunlight, but there is a twist. There are more than just one type. Plants have four main pigments. The first is chlorophyll A, which is bright blue-green and is the main pigment for photosynthesis. The second is chlorophyll B, which is yellow-green and is an assistant to chlorophyll A. Third is xanthophyll, which is yellow pigment. Fourth is carotenoids, which is yellow-orange pigments. Each pigment absorbs light best at specific wavelengths. Chlorophyll A is the chief pigment, absorbing mainly blue and red light, the most effective colors for photosynthesis. But chlorophyll does not work alone. Accessory pigments like chlorophyll B, xanthophylls, and carotenoids catch other colors of light and pass that energy to chlorophyll A, making photosynthesis more efficient. These pigments also protect chlorophyll A from photooxidation, which basically is damage from too much sunlight. The first stage is light reaction, a photochemical phase. It happens in the thylakoid membranes, the stacked discs called grana. Here, the pigments are organized into light harvesting compounds or LHCs, which belong to two systems, photosystem 1 and photosystem 2. In photosystem 1, the reaction center is P700. It absorbs red light at 700 nanometer. In photosystem 2, the reaction center is P680. It absorbs light at 680 nanometer. These pigments work together to trap sunlight and pass that energy to special chlorophyll A molecules. In photosystem 2, light excites electrons at P680, boosting them to a higher energy level. They are passed to a primary electron acceptor and then travel downhill through an electron transport chain. Meanwhile, in photosystem 1, P700 also absorbs light, exciting its electrons, which are passed to another acceptor and finally used to reduce NADP plus into NADPH-, an energy-rich molecule. This step-by-step energy climb and drop is called the Z scheme, the pathway of electrons from water in PS2 to NADPH in PS1. In this process, light first boosts electrons in PS2 to higher energy level. They lose some energy as they move through the transport chain, and then light in PS1 re-energizes them so they can be used to make NADPH. But where does PS2 get its replacement electrons? It's from splitting water, a process called photolysis. Water breaks into electrons to replace lost ones in PS2, protons or H+, used later for ATP formation, and oxygen released into the atmosphere. In non-cyclic photophosphorylation, both photosystem 2 and photosystem 1 work together in sequence. Light hits photosystem 2, exciting electrons in P680. These high-energy electrons travel through an electron transport chain, releasing energy to pump protons across the thylakoid membranes. This energy drives ATP synthesis. The electrons then reach photosystem 1, where another photon of light re-energizes them in P700. Finally, they are passed to NADP+, forming NADPH. The electrons lost from PS2 are replaced by splitting water, producing oxygen as a byproduct. In cyclic photophosphorylation, only photosystem 1 is active. Light excites in P700 and instead of moving to NADP+, they loop back through the electron transport chain, returning to photosystem 1. This loop pumps protons just like before, producing ATP to balance the energy needs of the Calvin cycle. So how does a light reaction actually produce ATP? The answer lies in the chemiosmotic hypothesis. As excited electrons travel from photosystem 2 through the electron transport chain towards photosystem 1, they don't just carry energy for making NADPH, they also power proton pumps embedded in the thylakoid membrane. This pumping creates a proton gradient, a high concentration outside the stroma. It's like charging up a battery, storing potential energy across the membrane. Now the gradient wants to balance out. The protons want to flow back into the stroma, but the only way through is via a special enzyme called ATP synthase. As the proteins rush through the ATP synthase, like water spinning the blades of a hydroelectric turbine, the enzyme harnesses this flow to join ADP with inorganic phosphate, producing ATP. The ATP, together with the NADPH made in the light reaction, will power the next stage of photosynthesis, the Calvin cycle, where sugars are built. We have just seen how the light reactions make ATP and NADPH. But where do these energy-rich molecules go? They are the fuel for the next phase of photosynthesis, the biosynthetic phase, also known as the Calvin cycle. This set of reactions happens in the stroma of the chloroplast and is responsible for turning carbon dioxide into sugars. These steps are sometimes called dark reactions, but that doesn't mean they only happen in the dark. They can occur in the light or dark, but they absolutely depend on the ATP and NADPH from the light reactions. The Calvin cycle starts when carbon dioxide enters the stroma and meets its partner, the primary carbon dioxide acceptor. Scientists once thought this acceptor would be a 2-carbon compound, but experiments revealed the truth. It's actually a 5-carbon sugar called ribulose 1,5-biphosphate or RUBP. When RuBP combines with carbon dioxide, the reaction is catalyzed by the enzyme Rubisco, the most abundant enzyme on Earth. This produces two molecules of a 3-carbon compound called 3-phosphoglyceric acid. The Calvin cycle operates in three main stages: carboxylation, reduction, and regeneration. In carboxylation, carbon dioxide is fixed to RuBP by Rubisco, producing 3-GPa, the first stable product. In reduction, ATP provides energy and NADPH provides electrons to turn 3-PGA into a higher energy sugar molecule called triose phosphate. In regeneration, part of the triose phosphate is used to rebuild RuBP so the cycle can continue. This step requires more ATP. It takes 6 turns of the cycle, using 6 carbon dioxide molecules, 18 ATP and 12 NADPH to make just one glucose molecule. Some plants like maize, sorghum and sugarcane can photosynthesize under intense sunlight and heat much better than normal plants. That's because they use a special method called C4 pathway. Unlike ordinary C3 plants, C4 plants are adapted to hot, dry environments. They grow faster, waste less energy and produce more biomass. But how? Let's look inside the leaf. C4 leaves have a unique structure called Kranz anatomy. Kranz means wreath. Here the outer mesophyll cells surround tightly packed bundle sheath cells. Both cell types work together in 2 steps to fix carbon efficiently. Step 1. Mesophyll cells. In the mesophyll, the primary carbon dioxide acceptor isn't RuBP like in C3 plants. Instead, it's a 3-carbon compound called phosphoenol pyruvate or PEP. With the help of the enzyme PEP carboxylase, carbon dioxide turns PEP to form a 4-carbon acid called oxaloacetic acid or OAA. This 4-carbon acid travels into the bundle sheath cells where it breaks down to release carbon dioxide and a 3-carbon compound. The released carbon dioxide now enters the Calvin cycle just like in C3 plants to produce sugars. So the C4 pathway shuttles carbon in a clever loop. Mesophyll cells capture carbon dioxide in a 4-carbon acid. Bundle sheath cells release carbon dioxide right where the Calvin cycle operates. The leftover 3-carbon compound regenerates PEP, ready to start again. By concentrating carbon dioxide in the bundle sheath cells, C4 plants avoid photorespiration, the wasteful process that plagues C3 plants. This makes them more efficient, especially in hot, sunny and tropical conditions. Plants don't always use sunlight perfectly. Sometimes they make a big mistake, a process called photorespiration. Normally, the enzyme Rubisco fixes carbon dioxide to RuBP, producing 2 molecules of 3-PGA which go on to make sugars. But here's the catch. Rubisco can bind to both carbon dioxide and oxygen. When carbon dioxide levels are low or oxygen is high, Rubisco mistakenly uses oxygen instead of carbon dioxide. Instead of making 2 useful sugars, the reaction produces one 3-carbon PGA and one 2-carbon phosphoglycolate. This pathway is called photorespiration. And here's the bad news. In photorespiration, there's no sugar mode, no ATP produced. In fact, the plant actually uses up energy and even releases carbon dioxide. For C3 plants, photorespiration is wasteful. It lowers efficiency, especially under bright light, high temperatures or water stress. But C4 plants have a clever solution. They pump extra carbon dioxide into the bundle sheath cells, ensuring Rubisco only binds with carbon dioxide and not oxygen. That's why C4 plants have almost no photorespiration, making them far more productive in hot, sunny environments. Photosynthesis may seem simple. Plants use sunlight, water and carbon dioxide to make food. But in reality, the rate of photosynthesis depends on several internal and external factors. Internal factors. Inside the plant, things like the number, size and age of leaves, the amount of chlorophyll and even the internal carbon dioxide concentration affect how well photosynthesis works. A young, healthy plant with many green leaves will photosynthesize much faster than an old yellowing one. External factors. Light. Light is the most obvious factor. At low light intensity, the rate of photosynthesis increases as light increases. But after a point, more light doesn't help. The curve flattens because another factor becomes limiting. Interestingly, photosynthesis reaches light saturation at just about 10% of the full sunlight. So for most plants in open fields, light isn't usually limiting. Carbon dioxide concentration. Carbon dioxide is often the major limiting factor since its concentration in air is very low, only about 0.03 to 0.04%. Raising carbon dioxide levels up to about 0.05% can boost photosynthesis, but beyond that it can be harmful. C3 and C4 plants respond differently. C4 plants reach saturation at around 360 parts per million. C3 plants respond up to 450 parts per million or more, meaning current atmospheric carbon dioxide is still limiting for them. That's why greenhouse farmers often enrich the air with carbon dioxide for crops like tomatoes and bell peppers to increase their yield. Temperature. Since photosynthesis involves enzymes, it's temperature sensitive. C4 plants thrive at higher temperatures while C3 plants prefer cooler ranges. For example, C3 plants have an optimum around 20 to 25 degrees Celsius. C4 plants perform best at 30 to 40 degrees Celsius or more. That's why tropical grasses like maize grow well in hot climates, while temperature crops like wheat prefer cooler weather. Water. Water is essential, but not because it's used up directly in large amounts. Instead, water stress makes plants close their stomata, cutting off carbon dioxide entry. It also causes leaves to wilt, reducing their surface area and photosynthetic capacity. And that's photosynthesis. From the light reactions that capture energy, to the Calvin cycle that builds sugars, to the special C4 pathway and the factors that shape it all, is a process that powers nearly every living thing on Earth, turning sunlight into the food and oxygen that we depend on. If you found this journey helpful, don't forget to like, subscribe, and share to keep learning with me. Until next time, stay curious and remember the power of life begins with a leaf.